For decades, we thought we knew exactly how the brain gets built. The story was simple, it was elegant, and, well, as it turns out, it was pretty much wrong. Today, we are diving into a scientific revolution that has completely redrawn the genetic blueprint of the human brain.
Okay, so let's get right into it. For the longest time, the thinking was that building a brain was like building a pyramid. You had a few pharaoh genes at the very top, barking out all the orders. But the new reality? It's much more like a massive, bustling city where hundreds of different specialists have to coordinate and work together perfectly. We're going to explore exactly how we made this incredible jump in our understanding.
But, to really appreciate just how big a deal this new picture is, we first have to understand the old one. That comfortable, easy-to-grasp myth of the master gene that really dominated neuroscience for years. So, back in the 90s and into the 2000s, the idea was pretty straightforward. Scientists found a few superstar genes. You might have heard of them, like SOX2 and PAX6, and they figured these guys were the master regulators. The whole model was just a simple cascade. These top genes switch on, and then everything else just falls into line, one by one, like a set of dominoes.
And this quote from the research, it just nails that old way of thinking. A hierarchical cascade. See? It was neat, it was a top-down command structure, it made a lot of sense, it was easy to draw on a whiteboard, but, as we're about to find out, it was just a tiny, tiny piece of the real story.
So what happened? What just completely blew that old simple model apart? Well, a revolutionary genetic tool came along that let scientists stop looking just at the usual suspects and instead start investigating every single player on the field. We hear about CRISPR all the time for gene editing, right? But here, think of it as the ultimate diagnostic tool. It gave scientists this incredible ability to do something that was science fiction just a short time ago, go through the entire human genome, all 20,000 plus genes, and, in developing cells, just switch them off one by one to see what happens. It's like taking out every single spark plug from a car one at a time to figure out which ones are absolutely essential for the engine to run. And well, when the results from these massive genome-wide screens started pouring in, it became so clear that the master gene model just couldn't hold up. It didn't explain what they were seeing at all. They weren't finding a simple pyramid with a couple of bosses at the top. They found something much, much bigger.
All right, so let's shift gears and look at this new reality, at what this amazing technology actually uncovered about the brain's true architects. This is really where our understanding takes a wild turn.
This was the billion-dollar question, right? I mean, if it wasn't just a handful of these master genes, then how many were truly non-negotiable? How many genes do you absolutely have to build the most complex object in the known universe? A dozen? 50, maybe? The answer was just staggering. Get this, 331. Not 3, not 30, but 331 genes are absolutely indispensable. If you lose any single one of them, the whole process grinds to a halt. I mean, this finding was a total shock to the field.
So what does this all mean? It means the brain isn't built by a single pharaoh shouting orders from the top of a pyramid. No way. It's built by a distributed network, a highly coordinated population where hundreds of members are all critical for the project to succeed. We're talking collaboration on an absolutely massive scale.
And here's the really cool part. These 331 genes aren't just some random mob. They are a team of specialists. You got your project managers. Those are the genes that regulate what other genes are doing. You have the construction crew, the genes managing the cell's actual physical structure. You've got the power grid handling all the energy. And of course, the communications network for how cells talk to each other. Every single one of these teams is essential.
Okay, you might be wondering, how can they be so sure about all this? I mean, it's a huge claim. Well, scientists didn't just run one test and call it a day. They followed a super rigorous 4-step process to map this blueprint and make sure their revolutionary findings were solid. The scientific method here was just beautiful. First, they did that massive CRISPR screen to find all the potential candidates. Then, they took those 331 genes and classified them into those teams we just talked about. Then, and this is so crucial, they validated their findings using a bunch of different experiments to be absolutely sure they were right. And only then did they start applying this incredible new map to real-world problems.
Now this is where it gets really important, because this is way more than just some fascinating update for a biology textbook. This new, high-resolution blueprint of how the brain develops has profound, immediate implications for understanding and hopefully one day treating human disease.
And here is the real-world connection, the big so what.
When scientists compared their new list of 331 essential genes to the genes that are already linked with neurodevelopmental conditions, like autism spectrum disorder, they found a huge overlap. For the very first time, we have a map that might show us exactly where in the construction process things can go wrong.
So this map is not just a pretty picture, it's a practical guide. It's allowing researchers to build much more accurate disease models right there in the lab. It helps them screen for drugs that might fix very specific problems, and ultimately it creates a real roadmap for precision medicine, for understanding not just that something went wrong, but precisely what part of this intricate genetic network is failing.
So we'll leave you with this thought. We've gone from thinking about a simple pyramid to understanding a complex, interconnected city when it comes to the brain. And that just forces us to ask a bigger question. Now that we see this beautiful complexity here, what other fundamental parts of our own biology are we still oversimplifying, just waiting for the right tool to come along and reveal their true amazing nature?